Okay, this is revision video 4B2. I'm just going to go through all the hot topics list, the grid that you would normally get, as you can see here on the screen. I'm just going to go through each point just as a quick revision recap. Okay, so the first thing we think that might come up is the process of asexual reproduction. All right, this is how single-celled organisms will reproduce. So they'll just copy themselves because they're a single cell big. So they'll just copy themselves, and the process with which they'll do that will be mitosis. You can see here on the diagram. Okay, the important thing here is when something reproduces by asexual reproduction, there's only one parent involved. So that could be, like I've said, a bacterial cell, like a single-celled organism. It could be a plant, so like a spider plant, strawberries, potatoes. They all reproduce asexually. So it's one parent. So the offspring will be genetically identical, and it will be described as a clone. Okay, important words for you to be able to use when describing asexual reproduction. The second thing then we think that will probably come up, because it always comes up, is being able to draw a plant and an animal cell. These are the key features here. Okay, you know that both have a cell membrane, but a plant cell has a cell wall, an animal cell does not. Both have a nucleus, which is where you'll find the chromosomes, so the DNA of the animal or the plant. Both have cytoplasm, that's the jelly-like substance in the middle where most reactions happen. And the other additional features then, you've got ribosomes and mitochondria in both. Ribosomes are where proteins are made. Mitochondria is where the site of respiration, so where energy is produced. Plant cells also have a vacuole, a permanent vacuole, I'll try and use that word, and that's where cell sap is stored. So when it fills up, it pushes on the outside of that cell and allows that cell to become rigid or turgid to help hold that plant upright. The other thing is the plant cell will contain a chloroplasts, which contain the molecule or the chemical chlorophyll, which will absorb light for photosynthesis. Okay, so please make sure you know the differences between the two and you're able to label them. Right, the role of the organs in the digestive system. This is going to come up later as well in just like the list of them, but then we need to go through the specific parts that each thing does. So in the mouth is where it starts with mechanical digestion, so breaking down the food using your teeth. So that's then swallowed, travelling down the esophagus, and you can see that's labelled down the diagram. So it travels down the esophagus into the stomach. In the stomach is hydrochloric acid. Please don't forget the name of the acid is hydrochloric acid, and it means it's a very acidic environment, so pH 1 or 2. In there is some enzymes. Protease enzyme is the one that's present in your stomach. So that will not only churn, because remember the stomach contains muscular tissue as well as glandular tissue. So glandular tissue secretes the enzymes and the acid, and the muscular tissue will churn. Okay, so it's a movement as well as chemical digestion occurring. That then gets squirted out into small intestine. The small intestine absorbs all the food out into the bloodstream, and then it travels down into the large intestine, and the large intestine will absorb the water, leaving behind the feces, will then be excreted. A couple of other features involved in that as it travels through is the liver. The liver sits just above the intestines, as you can see on the diagram. Basically, that produces a chemical called bile. Bile is stored in the gallbladder, which again is labelled on the diagram. And basically, bile sits in there until it's secreted into the intestines. And what bile does is it neutralises the acid coming out of the stomach, so it doesn't damage the intestines, but it also helps emulsify fats. So bile has two processes that it gets involved in: neutralisation of the acid and emulsifying the fats. Okay, so that's the organs of the digestive system and their role. Okay, we also think explaining why muscles store glycogen will come up. So remember, glucose is stored as glycogen in animals. Plants store it as starch, but animals store it as glycogen, sorry. Glycogen, therefore, can be broken down when we need it. And why do we need it to be broken down to glucose? Well, we need it for respiration. Okay, so respiration is glucose with oxygen converting into carbon dioxide and water, but releasing loads and loads of energy. So we store glucose as glycogen in the muscles, because the muscles is where we're going to need a lot of energy to be produced for movement, etc. So we store it as glycogen, and then when we need it, we can convert it back into glucose for respiration. Okay. Commonly, you'll get a question on catalysts or enzymes. Remember, please, that a catalyst increases the rate of reaction. You know this from chemistry, and basically an enzyme is a biological catalyst. Okay. Right, going on to the next one then. 3 limiting factors of photosynthesis. You must be able to draw and label and explain these graphs. So the first one is light intensity and photosynthesis. So what you can see on this one is if you look at the bottom axes and use the words on the axes to describe it, as light intensity increases, so does the rate of photosynthesis. So you can see that graph increasing. Then it starts to plateau and level off, and that's because something else has now become a limiting factor. So it could be the level of carbon dioxide. So although you've got increasing light going ahead, actually you haven't got enough carbon dioxide, so that's now limiting the rate of photosynthesis and it's remained stable. The graph for carbon dioxide concentration looks exactly the same. So again, as you increase carbon dioxide, the rate of photosynthesis is going to keep increasing. However, something else is going to limit it and it's going to go flat. Okay, so again, in that case, it could be light intensity. Temperature looks slightly different. Temperature increases and then drops off. So increasing rapidly and then decreasing rapidly. And the reason for that is as you increase temperature, the particles will move more, so you'll get an increase in the rate of photosynthesis. However, you will reach a point in temperature where those enzymes involved in photosynthesis will become denatured. So it will drop off. Okay, so the rate of photosynthesis will then decrease because the enzymes have become denatured. Right, onto the second column. This one is then explain why cells differentiate. Cells differentiate to become different cells. So we start with a ball of stem cells and they differentiate and become different. So like I said in the revision video before, you are all stem cells. You are going to become different when you leave school, college, university. You're going to get jobs. At the minute you do not have jobs, you don't have a role, you're stem cells. So stem cells are unspecialized cells. They then differentiate to become specialized cells as we grow. And in a plant it does exactly the same thing. And you'll find the stem cells at the shoot and the root where the plant is growing, and they will become specialized as they become stem cells, and they'll specialize into different cells in a process called differentiation. Okay, on to the next bit then. So describe how to carry out a line transect. Very common 6-mark question. Nice method here to be able to write down. So we would use a line transect when we are looking at an environment that changes. So for example, this diagram here, you can see there's a watery area. So in terms of that, it's labelled tidal creek. And then as you go up the bank, you can see that the plant life is changing. This would be a perfect situation for a line transect. So what you would do is you'd put down a rope or tape line, and this can be any distance. I've said 20 meters here, but you could do it for longer or for shorter. And then what you do is you use your quadrat, that square grid, and you'd place it about every metre along that line, and that's a transect. It does need to be at set intervals, so you can make sure it's fair test, so keeping that interval the same as you go up. And all you would do is you would measure and record what you see in the quadrat in terms of plant species. So you might find that a species you find very close to the river actually isn't present as you go further up the bank. So line transects are used where the environment is changing rapidly. Right, describe the process of anaerobic respiration. Anaerobic respiration is respiration in the no oxygen, no lack of oxygen. So here we have glucose, no oxygen, so just glucose going to make lactic acid and some energy. You do get energy being produced, but it's far, far less than aerobic respiration. This would be occurring in instances where you have a burst event, so weightlifting, sprinting, etc. Right, 3 different plant tissues and their functions. So 3 tissues you need to be aware of as epidermal tissues, which you can see here in the diagram is that top and that bottom part. It's the layer which covers the plant. The mesophyll is another type of tissue. The mesophyll, again, you can see in the diagram, is made up of 2 types. It's the palisade and the spongy. Palisade cells are cells that are more rectangular and uniform and tightly packed. There's loads of them, and they contain lots and lots of chloroplasts because they're near the top of the leaf for photosynthesis. The spongy mesophyll are more circular and they're spread out so that air can get in and out. So we need carbon dioxide to come in for photosynthesis and oxygen to leave as a byproduct. Also, the other 2 types of tissue would be your xylem and your phloem. They are responsible for transporting things up and down the plant. Remember, the xylem are transporting water and the phloem is transporting sugars. So they are 4 plant tissues. Again, you must know the names of them and be able to recall them. Right, explain the oxygen debt. So oxygen debt would be when somebody is going through anaerobic respiration and producing loads and loads of lactic acid because they're respiring without oxygen. So again, you can see here it's that burst event, that sprinting, that weightlifting. The lactic acid is what will build up, and the only way we can break that down is by using oxygen. So when you finish that burst event, you breathe deeply and rapidly, and that oxygen is going to be used, first of all, to break down lactic acid before you can start respiring aerobically again. So what we're doing is we're using the oxygen to pay off that debt we have that we're owing, so it breaks down lactic acid, and then we can start respiring aerobically. So oxygen debt is using the oxygen to pay off or break down lactic acid that we've built up because we were respiring anaerobically. Right, 6 different causes of extinction, and be able to list them. Again, commonly comes up, please make sure you know them. So we can have changes to the environment. If the environment changes, it's likely that we could have a case of extinction, and this is over a huge time period. New predators. New predator comes in that the animal can't compete with, can't get away from. The predators can catch it far quickly and use it as prey. That's going to be a problem. New diseases that they haven't become immune to over time, so there's no immunity there, so they'd wipe out a species, or could wipe out a species quite quickly and make them extinct. New, more successful competitors. So this is when something else will be competing for your food source. It's not hunting you, it's going for the same food source you are. So you are now going to be lacking nutrients and therefore over time become extinct. A single catastrophic event could also make a species extinct. This could be the volcano, so ice age, etc., collisions with asteroids. And also just through the cyclic nature of speciation, new species arising and then species becoming extinct. Okay, so 6 clear causes of extinction, and they are lifted directly from your specification. So again, please make sure you can recall them. Right, draw and label the parts of the digestive system. We've looked at this already, so it'd be worth just maybe pausing it, covering it up, see if you can label it, or see if you can recall those labels on that diagram. So starting with the mouth, through the esophagus, down into the stomach, you've got your liver and your gallbladder at that stage as well, and then you've got your small intestine, which is the part in the middle, tightly packed, the large intestine going around the outside, okay, and that's the area, the organs of the digestive system. So we've already touched on this one. Explain when cells differentiate. So cells differentiate in our body to give rise to different cells. And that occurs when we are at an embryo stage. So the sperm and the egg join together and fertilise and form a zygote. That's that first cell of a baby. And then that starts to divide by mitosis, remember, forming genetically identical cells. And so we've now got a ball of stem cells. Those stem cells will then differentiate as the embryo is becoming a fetus. They're becoming different. So they might become, like on the diagram, red blood cells, muscle cells, brain cells, sperm cells, heart cells. They're all becoming different. So cells differentiate during that growth period. Okay. In a plant, however, the plant retains more ability to differentiate throughout life. We don't as animals, but plants do. So again, that's a subtle difference there in terms of differentiation. Okay, drawing a yeast cell. Yeast cell looks like the diagram here. So we've got cell wall and cell membrane, cytoplasm and a nucleus. We will look at bacterial cell in a bit. Remember, that doesn't have the nucleus. The yeast cell does have a nucleus. Okay, define the term gene. So a gene is a unit of inheritance. It's a thing we inherit from our parents that give us our characteristics that we can see. They're found on our chromosomes. So if you see here on this diagram, you can see the X shape, the top, which is a chromosome. That's made up of tightly packed DNA. If we look at just a small section of that DNA, that would be a gene. And that gene basically codes for a protein, and the protein will then give us our characteristics. So it might be coding for the protein, which gives me the pigment to get my eye colour, okay, or the hair colour. So they're a unit of inheritance. They're found on our chromosomes, and it's a section of DNA that codes for a protein. Please, please, please remember size order. DNA the smallest, then gene, then chromosome. Really important. Okay, next one. Define protein and give 4 main uses. So what do proteins act as? So the structural components of muscles with different tissues, but muscles would be an example. They could be hormones, they could be antibodies, and they could be catalysts or biological catalysts, enzymes. Okay, so structural components of tissues, hormones, antibodies, enzymes. That's what proteins are. So remember that enzymes are proteins, so they become denatured by those high temperatures. Next one, explain what happens to glucose produced in photosynthesis. So why does the plant bother making its own food? This glucose, what does it use it for? Well, it could use it to produce fats or oils for storage. Could use it to make cellulose, which strengthens that cell wall. Remember, it's got a cell wall and then a cell membrane in a plant cell. So it's a cellulose cell wall, makes it much stronger. It could also be used to produce proteins, but remember we do need nitrates, and nitrates come from the soil to make protein. But it could also be used in respiration. So remember that plants respire just as they do photosynthesize. So any part of the plant that isn't green, so it doesn't contain chlorophyll, will have to create energy via respiration. So remember that they could use the glucose they make in photosynthesis and then use that in respiration to create energy. Okay, on to column 4. So explain fertilization. Fertilization is the joining of the male and female gamete. So in humans, it's the sperm and the egg cell. Remember that the sperm and the egg cell are what we could describe as haploid. They contain half the genetic information. So for us, our body cells contain 46 chromosomes. The sperm and the egg will contain 23 each. So when they join together or fertilize, they'll contain 46. So fertilization is when a male and a female gamete join together and fuse together to form, in this case, you've got a zygote being formed. Okay, explain why microorganisms are used to make enzymes. Well, enzymes increase the rate of reaction for industry, so it's very, very useful. So we can use microorganisms, we can use their enzymes because it increases the rate of reaction, and therefore if you're in industry, that increases your amount of product and therefore profit. They're not used up in any way, so that's really, really good. That's the enzyme, they're not used up, and they're used for lots of different things. So examples of how they are used might be yogurt, cheese. We've also looked at baby food, biological washing powders, etc. for uses of enzymes. Okay, summarise the issues surrounding stem cells. This is a key question where you don't want to start being waffly and going on and on and on. You want to try and keep it concise. So some arguments might be that to use stem cells, particularly for animals, we know that the best type of stem cell, the stem cell that's most potent, is how we describe it, the one that can turn into anything, would be embryo stem cells. Now, embryos are going to develop into a baby. So if we use those stem cells, we've effectively aborted that child. Some might describe it as murder. Okay, we've given it no choice. So it's not had a choice at all in terms of its embryo. And we could be described as playing God. Positive things, though, might be that we are furthering our knowledge of science for the benefit of everybody. So that would be a reason why I might want to do it. And obviously, as we've already talked about, treatments for disease like paralysis. So we may be able to grow nerve cells to transplant into somebody who's suffering from paralysis, or things like Parkinson's as well, using stem cells there trying to treat patients who are suffering from Parkinson's. So there are clear fors and against. And again, it's worth just remembering a couple of points so you can write those down in your exam. Okay, next one, describe the role of enzymes in detergents. So you should be thinking biological washing powders. So non-bio do not contain enzymes, biological washing powders do. So they might contain proteases to break down protein containing food. They might also contain lipases, and that breaks down any fats that you might have on your clothes. So protease and lipase will be present in detergents. And the reason we do this is because that detergent then becomes far more effective at a lower temperature. So if you're washing your clothes at a lower temperature, you are saving money because you're not having to heat up that water to such a high temperature. So again, that's a clear advantage in terms of using biological washing powders. The disadvantage is that some people might be allergic, which is why there are non-bio washing powders out there as well. We'll explain why muscles become fatigued during vigorous exercise. We've already touched on this. The idea of fatigue is due to lactic acid. So once we start respiring anaerobically, so we start exercising, we're breathing faster and deeper, and we have lots of oxygen coming into our blood, diffusing into our capillaries and then off to our muscles, and we're carrying out respiration. However, we then run out of that oxygen, and then we start respiring anaerobically, and that builds up lactic acid, and lactic acid is what causes fatigue, muscle fatigue. Okay, so muscle fatigue is the buildup of lactic acid in the muscles, and then any blood flowing through that carrying oxygen will then break that down. We'd referred to that already when we talked about oxygen debt. Okay, Right, name 3 different plant organs and their functions. So again, one that catches students out all the time because it's hard to remember plant organs compared to animal organs, which you're very familiar with. So the plant organs are stems, roots and leaves, and here are some descriptors you can see of what they do. So leaves obviously essential for carrying out photosynthesis. Remember that layer we talked about already at the start of this video, and the second layer down we've got the epidermal, and then we had that palisade mesophyll. They're packed full of cells with chloroplasts. Okay, so that can absorb the light of photosynthesis. The stem is obviously there for support, and the roots will take up that water needed in photosynthesis and minerals. And we've already said about nitrates being used by the plant to make proteins. So a couple of clear things there with organs and why we need them. Okay, define stem cells. Again, we've already touched on this. Stem cells are unspecialized cells, cells without a role, and they can grow into any type of cell in the body. Okay, we've got 2 types to remember. Adult stem cells, that's the cells that you'll find in you now, and they'll be in your bone marrow. They're not as good at dividing into anything, but they are a type of stem cell. And then you've also got embryonic stem cells, which is a type of stem cell which are more potent. They can turn into any type of cell. Okay, draw the structure of a bacterial cell. So key thing here to remember is that it has no nucleus. You get that big squiggly area, and that's just all the DNA loose inside the bacterial cell. So there's no nucleus in a bacterial cell. You've still got cytoplasm, you've got ribosomes, you've got a cell wall, cell membrane in the bacterial cell. You remember in the other video I've also said that you might get that circular DNA called a plasmid, and that is the bonus DNA that can be transferred from one bacteria to another, and that might contain information about how to be antibiotic resistant. So it might give the bacterial antibiotic resistance, so it can be passed on to other bacteria, either from one bacteria to another, or when that bacteria replicates, it will then have that antibiotic resistance, and that's in the circular DNA, the plasmid. Okay, describe the role of chromosomes. So chromosomes carry our genes, and here you can see what's called a karyotype. This is a picture of all of the chromosomes in one adult body cell. So you can see they're aligned in pairs because one comes from mum and one comes from dad. So if we pick chromosome number 5, you can see there are 2 lines. That's because chromosome number 5, you have one copy of chromosome number 5 from mum and one copy from dad, and they contain your genes. Okay, and those genes will then give you your characteristics to see what you look like and what proteins your body will be producing. So chromosomes carry our genetic information in the form of genes, and we have 46 or 22 pairs. And if you look at the final pair after 22, in this case it's an X and a Y, so this is male. If it was X and X, it would be female. Okay, explain the advantages of using enzymes in industry. Again, we've touched on this briefly as well. Enzymes are used to bring about reactions at normal temperatures and pressures, and if we didn't have them, it would mean that it would be quite expensive because we either have to increase the temperature or increase the pressure, all things that would cost us money. We might also have to get energy demanding equipment, which is more expensive. So effectively what we're doing is using it to lower the cost to the person producing it, and therefore lower the cost for the consumer. Okay, but the idea is that it's normal pressures, normal temperatures if we're using an enzyme. Okay, describe cystic fibrosis and how it's inherited. Cystic fibrosis is the recessive disease that you need to be aware of. Remember, the dominant inherited disease is polydactyly, which is where you've got an extra finger or toe. So cystic fibrosis is the example of the recessive inherited condition in your specification. Here you can see a diagram. So you can see that the mother and the father are both carriers of cystic fibrosis. They don't have it, but you can see again there, like we saw on the karyotype, they've got a pair of chromosomes from their mother and their father. One of them passed on cystic fibrosis and one of them didn't. So they're both carriers. And you can see in the picture that what that gave them is it gave one child who didn't have it, 2 children who could be carriers, and 1 child who would have it. So if they had a child, they had a 1 in 4 chance of having a child with cystic fibrosis, a 1 in chance of having a child who is completely unaffected, and a 50% chance of having a child who is a carrier. If that's drawn as a Punnett square, which is the box you can see at the bottom, if you just look at the top under mother, it's got a big C and little c, and all that does is that represents those alleles being inherited on those chromosomes. So those versions of the gene will come to allele towards the end of this revision video, what that means. And I'll show you this diagram again then. So the mother has got a big C, so a healthy copy of that gene, and then a little c. The little c is the one that codes for cystic fibrosis. But because she has a big C, she doesn't have it because the dominant takes over, and the father is exactly the same. And then you literally just draw it into the box. So you put the letters in as they show underneath, and there you can see the ratios coming out of the side. Okay, explain how fossils are formed and their importance in evolution. So fossils are essential in evolution to describe and show us what has happened. So it's our only evidence given to early life forms. That's really all we have to go on in terms of showing how the bone structure, etc., has changed over millions of years. So they are vital in showing any sort of evolutionary links between different animals. They can be formed from the hard parts of animals that don't decay easily. They can be formed because parts haven't decayed because the conditions we needed for decay are absent, which might mean being warm, having moisture, etc., and having oxygen. They're all needed in order for something to decay. So if any of those are lacking, then we may find something quite intact. We may find that parts have been replaced by other substances. So, for example, the hard parts have decayed slower, but they're still decayed. And they might be squashed in between two rocks, and then some water from a river carrying some sediment has got in between, and that sediment over time has filled that gap and gone hard. And so that that's what's uncovered from the fossil. Then you also may get preserved traces like burrows or footprints as well. So they are the different ways in which fossils can be formed. Okay, on to the final column then of things we think might come up in your exam for B2. So describe how to carry out a random sample of organisms. The word random, you should be thinking quadrat, okay, not line transect. If it's completely random, it's using your quadrat. And this is where you do it on a uniform area, so maybe the playing field, for example. And you would put your quadrat down randomly, and a way to do that might be to close your eyes when you throw it. That would be a fine answer to give for your exam. Okay, place it down, and then you'd record what you can see in each square. Remember, it's worth working out what the percentages of each square. So count up how many squares you've got, divide it by 100 to find the percent of each square. Then you can work out the percentage cover in the quadrat. Okay, explain how sexual reproduction gives rise to variation. Variation just means different. It means varied. We're all different. So even the children from the same two parents will be different. And the reason being is because sexual reproduction involves a male and a female gamete coming together and forming a separate individual. So because we've got that mixture of DNA, that is what's going to give rise to variation. So asexual reproduction, we talked right at the start, there's no variation whatsoever. It's a clone. It's an exact copy of that one parent. But sexual reproduction is the male and the female gamete joining together and forming that zygote, and then that will be different to the next one that's formed because that will be an entirely different sperm and an entirely different egg. Here you can see a picture to demonstrate that of some frogs all showing variation. They all have differences. Okay, so this is what we just mean by variation. Things are different, and it's because they are produced from sexual reproduction. Okay, describe the different uses of energy from respiration. So we know we produce energy in respiration, and we can use it to either build larger molecules from small ones, for muscles to contract, so for movement, for keeping a steady body temperature in colder surroundings. So when we're really cold, to keep us warm, we're going to need to use energy. And also to build up sugars or nitrates into amino acids. Okay, amino acids are the building blocks of that protein. Remember I said that we're making those amino acids, and they join together to form that protein. Okay, mitosis, explaining mitosis, bit of a harder one. Mitosis is when we produce a genetically identical daughter cell. So this is asexual reproduction. This is cloning, but in us, we would use it for growth and repair. So if you cut your skin, you produce an identical skin cell via mitosis. So we started off with one cell. Here we've got 2 chromosomes in it, if you have a look at the diagram. Then it's replicated. So chromosomes, if you remember back to the karyotype I showed, were just 2 lines. And that's what a chromosome is. It's not actually an X, it's just a line. It then replicates itself, so you can see them looking like an X shape. So basically one line has copied itself, but they've stayed stuck together. Those 2 chromosomes then now you can see are pulled apart from that centre point. They're yanked apart. So what you end up with is 2 cells with identical copies in at the end. Okay, so mitosis produces 2 genetically identical daughter cells, and that's the key part of information you want to take away. It's cloning. Okay, so it's growth and repair in us, or asexual reproduction. Give the equations for photosynthesis and respiration. You should know these by now. If you know one, you know the other because they're just backwards. So respiration is glucose. So we do this by eating, breathing in oxygen, and then producing carbon dioxide and water and loads of energy. Photosynthesis is literally that in reverse. Look at what the words say. You've got carbon dioxide and water coming into the plant plus your light energy, which will then give the plant glucose, which we'll then use in other things, which we've talked about already, and oxygen, which will be released out of the leaf. Okay, so that's your 2 equations. Okay, and finally for your B2 revision, we're looking at defining an allele. Okay, an allele is a version of a gene. So for example, if we have a gene which is hair colour, our versions would be blue, brown, black, for example. In this one here, you can see the diagram. Again, like I said before, when we looked at cystic fibrosis, I'd show you this slide again. With cystic fibrosis, you inherit, or with any gene, you inherit 2 alleles, 2 versions of that gene, one from mum, one from dad. In this case here, mum and dad are both carriers, so they have a healthy version, a healthy allele, which is the dominant, but they also have an allele which could give you cystic fibrosis if you inherited both small c's or both recessive alleles, both recessive versions. In this case, if you look at the diagram, the 2 that are both purple, both blue, the children, have again inherited as a carrier. So they've inherited a capital C, the dominant allele or dominant version, which makes them healthy. But they've also inherited the small c or the recessive allele that would make them both carriers. The completely purple girl is unaffected entirely, has not inherited any small c from either parent. She's inherited both alleles that are dominant, so she's completely unaffected. However, the blue boy, in this case, has inherited both small c's, okay, and therefore that child will have cystic fibrosis. Please make sure if you're doing cystic fibrosis and they give you c's to draw in the exam, like a genetic diagram, you make it so clear when you draw big C and little c. It's a rubbish letter to use, but they often still get you to use it. But please make sure you're so neat with your writing that you're very clear whether you want to do a large C or a small c, because that's an easy way to lose marks. Okay, so an allele is a version of a gene. Right, that is the end of the hot topics, the things we think might come up. So good luck for your exam, and I'm sure you'll do brilliantly.